कोई कुछ कर रहा है। ये सब ये बैठे हैं, कहाँ पर यहाँ पे बैठे हैं, मैं भी वो है।
तो इन लोग को पूछ लो एक बार।
यार can I use the cabin for the class if you people are okay with? हाँ, because वहाँ भी मैम पढ़ा रही है, ये भी बात कर रही है, मीटिंग ले रही है। हम यहाँ वो NPTEL वाला क्लास लेना चाहते थे, if possible। थैंक यू मैम। आज हमारे साथ ही मीटिंग कर रही थी, 7 बजे से स्टार्ट होना था।
अगर कुछ बात ना करें तो
नहीं कर
तुमने एक मिनट डिस्टर्ब होगा तो मैं जाऊँ
ना बैठ।
थोड़ा बंद कर ले हाथ
So hello everyone, we have waited for 5 minutes for someone else to join, but since no one else is joining, we'll go ahead with the presentation. I hope my screen is visible. So I welcome you all to the Bioengineering and Interface with Biology and Medicine NPTEL course, PMRF-NPTEL session, week 6. So PMRF-NPTEL sessions are interactive problem-solving sessions in which PMRF associated with the course, such as myself, takes these sessions with the students who have applied for the particular course and solves various assignment problems of that particular week in order for the students to have a better understanding and how exactly to face the assignment questions as well as the final exam, as well as have a better understanding of the course material. So this week we are going to discuss the week 6 of the NPTEL course Bioengineering, in which we learned a lot about cell cycle regulation, cell cycle dysregulation causing cancers, as well as a bit on developmental biology and animal cloning. So since no one else has joined the meet yet, we'll go ahead and we will go ahead with the session as is, and let anyone join who wants to at a later point. So let me introduce myself. My name is Anup Chatterjee. I'm a PMRF fellow and PhD scholar at the Developmental Neurobiology Lab, Department of Biological Sciences and Bioengineering, Indian Institute of Technology, Kanpur. So as I said, this week we have a lot to discuss based on cell cycle regulation as well as uh, parts of animal cloning. So we'll just go on and get ahead with it. Okay. So this is the first question of today. The question is, the cell cycle is a set of activities that take place inside cells to produce daughter cells by dividing the cell cytoplasm and nucleus. The cell cycle has four key stages that a cell goes through. In any eukaryote, what is the ideal stage for counting the total number of chromosomes? And also, at what point do chromosomes align on the equator of the spindle fiber? So as we know, during cell division, there are a lot of processes that occur inside the cell. A major part is what is known as karyokinesis, or the division of the nucleus. This occurs via division via various processes in which the chromosomes first condense and supercoil and are into sister chromatids. They are brought to the metaphase plate, and that is at the center of the cell, from which spindle fibers then separate the two sister chromatids as individual chromatids, and then cytokinesis follows this process, which essentially divides the cell into two parts. So our question is, amongst all of these steps, which is the most ideal stage for counting the total number of chromosomes? And also, at what point do chromosomes align on the equator of the spindle fiber? What is the stage known as? So we are asked to pick the correct combination for both of these. The first option is, the best phase to count is prophase, and equator alignment occurs during early anaphase. The second option is, best phase to count is metaphase, and equator alignment also occurs during metaphase. Third option is, best phase to count is anaphase, and equator alignment is in late anaphase. And the final option is, best phase is to count is telophase, and equator alignment occurs during the metaphase. So I would encourage everyone to try and think this through and come up with an answer. This is only one option, correct type of question. So only one of the options that are given here are correct. Please go ahead and try to answer this. Okay. So hello Rishita, we were actually solving one of the questions of week 6 of the NPTEL course. The question is as follows. You can see it on the slide. You can try and answer the question. Like put whatever you think is the correct option in the chat box after reading it. Okay. So we will go to the solution slide and try and understand this problem before and try coming back to it and answering it. So the entire cell cycle is a complicated set of procedures that occurs during the division of a particular cell into its two daughter cells, and it occurs in a sequential manner in a cycle. That is, the cells go through the entire process again and again till it has reached what is known as senescence. That is when the cell stops dividing. So let us first understand what exactly is the cell cycle. So a cell cycle is a series of events that takes place in a cell as it grows and divides to produce two daughter cells. A cell normally spends most of its time in what is known as an interphase, in which it is growing and it's preparing for cell division. It replicates its chromosomes and prepares for cell division to take place. This entire process is just preparation and no actual division of chromosomal material or the cytoplasm occurs during the interphase. And when the cell leaves the interphase, it undergoes mitosis, that is the division of the cell or cell division to form two daughter cells. So let us go into a bit detail on what exactly these processes are and what are the various phases of cell cycle. The first phase, as we can see right here from the diagram, is the G1 phase or the cell growth phase. So the growth phase of G1. During this particular phase, the cell grows in size, synthesizes various cell organelles required for the cell division, and other macromolecules including proteins which will help for the cell to divide. The cells also accumulate sufficient energy for the division to take place, as you would assume correctly that the entire cell division process is very energy requiring, and the G1 phase is when essentially the cells stock up on this energy in the form of ATP, which will later help to divide. Next phase is the synthesis phase. In this particular phase, the existing DNA is copied within the nucleus. So DNA duplication will occur during the synthesis phase only. And the centrosome, which is important organelle which is responsible for cell division, is also duplicated during this phase and gives rise to what is known as spindle fibers. We'll learn more about these spindle fibers in a moment, but centrosome is essentially the organelle that produces the spindle fibers, which attaches to various chromosomes. These chromosomes are then divided into the two daughter cells using these spindle fibers. We'll look into the role of them in a bit. So this occurs during synthesis phase, the copying or duplication of DNA and the duplication of centrosome. So this is the is shown in the diagram where DNA replication takes place. Third is the growth phase 2 or G2 phase, in which cell grows further in size, makes more proteins and organelles required for the entire mitosis process, and all preparation for mitosis gets completed. So this is the final stage at which the cell has prepared to undergo mitosis and every kind of checkpoints are present, which ensures the mitosis occurs. So there is another important point to the G1 and G2 phases. The G1 and G2 phases act as checkpoints in which in the both phases the amount of proteins, the energy requirement as well as correct duplication of DNA are all extensively regulated so as to make sure that the cell can undergo the cell division, that is it is completely prepared, after which there comes the M checkpoint which also checks all of these parameters and then the cell goes ahead into mitosis or cytokinesis. So the next stage is mitosis, the M phase and cytokinesis. It is the nuclear division period that consists of four phases: the prophase, metaphase, anaphase, and telophase. And during this phase, the cell divides the nucleus, that is karyokinesis occurs, and gets separated into two daughter cells, which is known as cytokinesis. And each of the daughter cells receives a complete set of chromosomes. So this is the mitosis phase, and finally there is another phase which is so the mitosis phase is summarized here, in which we have the prophase, metaphase, anaphase, and telophase pointed out, and finally there is cytokinesis that occurs that goes on simultaneously with the telophase. And then we have another phase that is essentially outside the cell cycle, which is known as G0 or growth phase 0. In this particular phase, the cell exits from the G1 growth phase and enters a dormant or resting state. It essentially ceases cell cycle and will not participate in cell cycle anymore till external cue is received. So, an important point to note here is that most somatic cells in our adult body are actually in G0 phase. These include neurons, nerve cells, and various other cells which have ceased any kind of replication anymore and will remain in the restive state for the rest of adulthood. So this was essentially a summarizing slide on cell cycle. If anyone has any questions, they can post in the chat or you can unmute yourself and ask anything. Yes, Rishita?
So, what is the significance of G2 phase, growth phase 2?
Okay, so the thing is that even though the names are very similar and we have not delved very deeply into what exactly the proteins that are being synthesized in both of these phases, these are vastly different proteins that get synthesized. So, growth phase 1 essentially stocks upon proteins that are necessary for the synthesis phase, and growth phase 2 stocks upon different proteins that are required for the mitosis itself. Further, there is a role of cyclins and CDKs, which actually regulate when the cell will go into the mitosis phase itself, that are being produced and degraded in the growth phase 1 and growth phase 2. So, since we are not dealing very deeply into this, this looks very similar, but the amount of DNA or amount of proteins, or the types of proteins that are produced in both of these phases are very different.
Okay. And sir, what is when the cell, how the cell knows that it has to enter G naught phase, or when cell enters into G naught phase?
Okay, so the first important there are multiple parameters that can be there. There are very signaling pathways that will inform the cell that it has to continue with its cell cycle or exist out of the cell cycle. A very important example that maybe you have you know of is known as contact inhibition. So normal cells they grow in a sheet type of layer where if the cell comes in contact with another cell, it has understood that there is no more space to grow and the cell stops dividing. That is when it is contacted by cells of all of these sides, it then receives a signal that it has to stop dividing and it exits the cell cycle and enters into the G0 phase. There are other parameters as well that occur here. Most of the cell divisions, like proliferative cell divisions, yeah, I will get to that, Sharon. So most of the proliferative cell division occur during the developmental phase of development of the embryo itself, but which is done by cells known as stem cells. But after the process known as differentiation, where these cells grow to mature cells such as which we know of, such as myocardiocytes, the muscle cells in our heart, nerves, bone cells. All of these, when they have completely differentiated to the final form, the cells do not replicate anymore. Our somatic cells, which are present in our body, do not receive any signals as such that allow them to replicate. If instead these cells are given various cues that help them to replicate, then that can be another thing. But in the when the entrance into the G0 phase is coupled, depends on these parameters: signaling from various cells that are around it, contact inhibition, as well as uh, the maturity or the differentiation state of the cell. So there are many parameters that are included in it. Is that okay, Rishita?
Okay, sir.
Okay.
Yes, sir.
Okay. So Sharan is asking what exactly is CDKs. So CDKs are cyclin dependent kinases. So another question that could have been there is that say we need, as we just talked about, what is the necessary condition for a cell to enter the G0 phase? But what is the necessary condition for the cell to enter the G1 phase or the S phase? When does the cell know that the G1 phase is over and the S phase has started? It should start DNA replication. Or when does it know that the S phase is over and the G2 phase has started? Or simultaneously, so on and so forth, that the G2 has ended and mitosis has started, or mitosis has ended and it has now to go into the G1 or G0 phase. What exactly regulates these? So there have been various experiments done, and scientists have found very interesting set of proteins which are known as cyclins, and another related protein which is known as cyclin dependent kinases or CDKs. What they have seen is that there is parallel or sequential growth in the expression of cyclins in one phase, and sequential growth of CDKs in one phase, and then degradation of the CDKs. So you see degradation of the cyclins themselves. So essentially, each of the phases are denoted by the presence of either the cyclins or the CDKs or both of them together. So if cyclin is present, it is one phase. If CDKs only are present, that is one phase, and if both of them are present, then that is another phase. So these are cell cycle regulators, which essentially tell the cell which phase it is currently in and what it has to do, which processes should it undergo. And these cyclins and cyclin dependent kinases are expressed sequentially and then also degraded sequentially as the cell goes through the entire cell cycle. Is that okay, Sharon? Hello?
Yes, sir, fine.
Yeah, okay. Thank you. So, if is there any more questions on this particular slide?
No, sir.
Okay. So we'll go forward. So this is the entire overview of the cell cycle. We will actually delve more deeply into the process of mitosis itself because it is way more important for our particular discussion, and we will know what exactly the importance of the different phases: the prophase, metaphase, anaphase, and telophase in the process of cell cycle. So, so we'll go forward. So this is what the mitosis looks like. So mitosis consists of four phases: prophase, metaphase, anaphase, and telophase. And the interphase, that is the part of the cell cycle in which the growth phase, synthesis phase, and G2 phase exists. So in the in the interphase, we have duplicated copies of DNA. So the the ploidy level is diploid or 2N, that is two sets of chromosomes. So then DNA is copied when the cell enters into the prophase, and in this particular prophase, the important thing that occurs is that the chromosomes pair up, or the chromosome are duplicated. So if in case of one deep blue strand of chromosome, we have two sister chromatids which are deep blue attached via the centromere. Similarly, we have instead of a light blue chromatid, we have two sister light blue chromatids that are joined at the centromere. So essentially, DNA is duplicated to form two chromatids for each of the chromosomes. Next, we have, after the prophase, We have metaphase, in which we have degradation of the nuclear membrane, and the chromosomes line up at the metaphase plate at the middle of the cell like this. So chromosome line up at the equator, and chromosomes line up at the equator, and they sit on what is known as mitotic spindles that we are just talking about. We'll go deeply into this. In the anaphase, what happens is that the spindles essentially pull the cytochromatids apart. So the centromere and the essentially the glue that is holding the chromatids in place are separated out, and we have two cystochromatids going to each end of the or the poles of the cells. So you have one set of chromosomes at one end. Finally, we have telophase, in which the chromatids have reached the particular poles, and cytokinesis that occurs simultaneously, in which the cell also starts dividing. So first we have nuclear divisions that occur. Nuclear material is being divided between the two ends of the cells. Then the cytoplasm will also divide, and which is known as cytokinesis. The cell pinches in the middle, and finally the pinching essentially is like a ball being squeezed in the middle. First it will elongate and then break into two, and it will form two identical daughter cells with one set of nuclear material, and the nucleus reforms to give us a two aneuploidy, like a diploid daughter cell, two diploid daughter cells from one diploid mother cell. So this is essentially the overview of mitosis. We'll go deeply into each of the phases now. So first, let us go through the first phase, which is prophase. So the main thing, what happens in prophase is that the cell destroys various organelles that it had, which are not necessary for cell division, or are there which are present in the interface and inhibit cell division. These organelles are destroyed, and then synthesizes other structures, which prepares it to undergo karyokinesis or nuclear division. So one of the important things that are destroyed is the nuclear membrane. So the nuclear membrane is destroyed, so we do not have any nucleus anymore. And other structures that are formed or synthesized or duplicated include the mitotic spindles on which the chromosome can attach to. So we have sister chromatids being formed, and essentially the pathways on which the sister chromatids can bind to being formed as well during prophase. So the chromosome supercoil to condense into sister chromatids. So we have a pair of sister chromatids for each chromosome. Mitotic spindle is synthesized by centrosomes. So centrosome is the organelle that forms these mitotic spindles that we can see right here. So centrosomes are these structures right here, and this is our mitotic spindles. Spindle fibers act as straps for the chromosomes to move. So for the chromosome, in order to move around in the cell itself, going to the metaphase plate or the equator of the cell, and then being pulled apart, it all occurs on a network of mitotic spindle. Essentially, the mitotic spindle act as railways for the chromosomes to walk on. So the chromosomes can only walk on these mitotic spindles themselves, and they actually are the ones that are controlling the movement of the chromosomes. Finally, the nucleolus disappears. That is, the nucleolus is a organelle which actually synthesizes ribosomes, and as well as the this is the sign which tells us the nucleus is getting ready to break down, and it completely disintegrates and releases the chromosomes into the cytoplasm for various next steps to occur. So this is what happens during.
So,
Yeah, Rishita, go ahead.
sir, as you have said that in prophase, the cell destroys other organelles. What are those organelles, and when are those organelles prepared in interphase?
Yeah, so one of the organelles is the nucleolus. So essentially, the nuclear membrane is destroyed. So many of these organelles are present, which are necessary for interphase to exist or the cell to not undergo the mitosis phase. So without the chromosomes actually being released into cytoplasm, they cannot travel to the equator and then be divided. So the first organelle that is being degraded is the nucleus itself. That is the first thing that gets degraded. And it is a symbol that the cell is not yet in the mitosis phase and it is still in the interphase. You are correct in that way, that these organelles signify that the cell is still in the interphase. And after these are degraded, that shows us that the cell is now moving towards the mitosis phase. Similarly, centrosomes are also not normally only one centrosome is present per cell. Duplication of the centrosome and formation of the mitotic spindle is also a signifying factor that shows us that the cell division is going to occur. Is that fine?
Yes, sir.
Okay. So, if no one has any other questions from this slide, then we can move forward. Okay. So, the next phase that is occurs is essentially a phase between the prophase and the metaphase, which is named as pro-metaphase, essentially in between two phases. It is also known as late prophase and sometimes called as pro-metaphase, in which the mitotic spindle begins to capture and organize the chromosome. So the mitotic spindle, so the nuclear envelope completely has broken down. The spindle go and attach to the chromosomes. The attachment point of the spindle and the chromosome is known as the kinetochore. The kinetochore is a protein complex which essentially guides where exactly the mitotic spindle will attach to the chromosome itself. So the chromosome become even more condensed, so they are very compact. So supercoiling occurs, and we have these sister chromatids, the X-like formation. So normally, as we know, that chromosome exists in a very decondensed form, where we have the stringy or string-like appearance of the entire DNA. But during cell division, they clump up to form very compact structures such as this. The nuclear envelope breaks down, so complete disintegration of the nucleus occurs, so that the cells are now exposed to the cytoplasm where the mitotic spindle can capture them, and this releases the chromosomes. The mitotic spindle also starts to grow more and expand throughout the cell, and some of the microtubules start to capture the chromosome. So essentially, these microtubules go and bind to the chromosomes. These microtubules that bind, so this particular one here that binds to a chromosome is known as a kinetochore microtubule. The ones that do not bind to the chromosome and are just attached to, from one centrosome to the other is a normal microtubule, a mitotic spindle fiber. These also have a role that we will just look at in a moment. So this is what occurs during prometaphase. So the next step is metaphase. So in metaphase, the spindle has captured all the chromosomes, so all the chromosomes are attached, and another thing occurs that they are lined up exactly at the middle of the equator of the cell. So they all the chromosomes which were just released into the cytoplasm, they were captured on the spindle fibers, which essentially is railway tracks, and they are all put directly in the middle of the cell. So we have all of them being aligned like very nice for people at the queuing at the middle or the equator of the cell. And this at the middle of the cell is what is shows us that the cell is at the metaphase. So this lining is something that is used to know at which stage the cell is, and also the fact that at this stage only the chromosomes are very well visible. So they are completely tightly condensed up, and they are present at one location only. So we can just go ahead and count them very easily at metaphase. So all the chromosomes align at the metaphase plate, and the metaphase plate is not a physical structure. It is not a physical structure, but just a term for the plane where the chromosomes lie. So if we draw a line through the middle of the cell on which the chromosomes are lining up, that is known as the metaphase plate, which is an imaginary line that we draw. Essentially, there is no physical structure that are holding them there, but that is just for our reference that we say that there is a plane that divides the cell in half where the chromosomes are present. At this stage, the two kinetochores of each chromosome should be attached to microtubules from opposite spindle poles. So what happens is that each sister chromatid has one kinetochore. So a spindle fiber goes and joins to this sister chromatid only, and another spindle fiber goes and joins to this sister chromatid only. So their attachment points are different and are present on either of the sister chromatids. So they are holding two parts of the chromatids by a two different centrosomes. So this will become apparent in the next phase, where they are pulled apart essentially by the microtubules contraction. And one sister chromatid will be pulled towards one centrosome and one pole, and the other towards another centrosome and another pole, which actually signifies the karyokinesis being occurring. Is that okay?
Yes, sir.
Okay. So next is the anaphase. In anaphase, each of the sister chromatid is pulled apart by the kinetochore to the poles. So anaphase occurs when in the metaphase they were attached to the microtubules on one ends, and now they are being pulled apart by the microtubules. So this is the pulling apart. So as you can see, each sister chromatid is now separate from the other, and they are being pulled to one end of the cell. Further, what happens is this occurs due to kinetochore microtubules pulling the chromosome towards the poles, the separation of the chromatids, and now there also comes the fact that the effect of the microtubules that were not attached to the chromatids. What they do is that they push at each other, so that there is a the microtubules start pushing at each other, and since the entire thing was circular to start with, when two ends start pushing at each other, the entire cell takes a elongated morphology. So we have an oval shaped cell from the previous circular cell. So the protein complex holding the sister chromatids together breaks down, forming individual chromosomes, and each pair of the chromosomes is pulled to the poles of the cell. So the protein complex is present at the sister chromatids breakdown, that is actually gluing the two chromatids together, breaks down. So we have two separate chromatids which are then pulled to either pole of the cell, and some microtubules that are not attached to the chromosomes, such as these ones, elongate to push the poles apart. And the cell then takes an elongated morphology or the oval shaped morphology. So this is what happens during anaphase. I guess that is fine. So that is what anaphase, what occurs during anaphase. And then finally we have the telophase. In telophase, the cell begins to return to a normal state. So normalcy to either parts. So now they have almost assumed in identity as individual cells, and normalcy starts to occur. And they go back to a state that was before the cell cycle started, and cytokinesis or division of cytoplasm starts to occur. So organelles and the parts of the cytoplasm get separated out into two cellular halves now, which signifies the end of the cell division. So the mitotic spindle are broken down. So as you can see, there are no more mitotic spindles present that are connecting the chromatids. The two nuclei are formed for each of the duplicated chromosomes. So the nuclear membrane starts reappearing around the chromatids, and a new set of nucleolus also forms. So this is the nucleus that is reforming. So essentially, they start re-establishing themselves as unique, identical daughter cells. So nucleus reappears, the nucleolus also reappears, and the centrosome, as you can see, now only one cell contains only one centrosome. That was what we started with before cell division started. And finally, the chromosomes uncoil to decondensed form. So from the sister chromatids that we saw, they start uncoiling to form the normal string-shaped DNA structure that normally they are normally occur in. So we have chromosomes starting to decondense, the nuclear membrane starting to deform, nucleolus reappearing, and the spindle fibers disappearing completely at the end of telophase. So telophase is also coupled with cytokinesis. So as you can see, division of cytoplasm is also now occurring, which occurs simultaneously with telophase, which is the last step to cell division. Any questions from this particular slide?
So,
Yeah.
as you have said that spindle fibers disappeared. So, do those spindle fibers dissolve in, or do they take another form kind of?
Yeah, so what happens is spindle fibers are formed out of monomers. So these are polymers of smaller subunits. The spindle fibers are essentially degraded to these monomers and then reutilized by the cell. So essentially they vanish out of the cytoplasm, they are degraded by the cellular structures and are reused in various other forms. So they just vanish. They do not change to anything else. So
And so
yeah.
one question that जैसे इंटरफेस में सेल अपने आप को प्रिपेयर करा होता है फॉर डिवीजन एंड वो अपना सभी अमाउंट जो डेवलप कर देता है सारी चीजों की डेवलप अमाउंट कर देता है इवन सेल ऑर्गेनेल्स भी डेवलप हो जाते हैं लेकिन प्रोफेज में वो सेल ऑर्गेनेल डिस्ट्रॉय हो जाते हैं तो वो पहले टाइम में बने ही क्यों थे बाद में
So what we have to understand is that the same cell organelles that are being formed in the growth phase 2, which are necessary for cell division, are not the ones that are being destroyed. Other cell organelles that were not necessary and were in hindering the cell cycle, which were not synthesized in any of the growth phases, were being destroyed. So these organelles include various things that are necessary for the cell to live in this interface only. So while it is in the interface, these organelles are present, such as the nucleolus, which is responsible for ribosome formation, which in turn is responsible for translation of proteins. But during cell division, none of this is required. So we do not need any new proteins being synthesized. Everything is already synthesized. And nuclear membrane essentially hinders the chromosome to walk into the cytoplasm and get divided. So these structures which are necessary in the interface for the cell to exist, but were not synthesized in the growth phases, are the ones that are being degraded. So these need to be present for the cell to live in the interface. Once they enter into mitosis, their usefulness is foregone and more necessary steps need to be taken, and then those organelles are degraded. The organelles being produced in the growth phases are the ones necessary for cell divisions to occur, and those remain there. They are not the ones that are degraded during prophase.
thank you so much.
Okay. So that was telophase. So next we have the final phase, which is cytokinesis or division of the cytoplasm. The division of the cytoplasm to form two daughter cells is known as cytokinesis, which occurs simultaneously with the final steps of mitosis. So it can, depending on the cell type or the cell type or how exactly the cell is, which stage the cell is in, it can occur during anaphase or telophase either. So in animal cell, a cleavage furrow forms. So as we can see right here, cleavage furrow is forming, which essentially occurs by the contraction of actin fibers. So actin or other cytoskeleton fibers are present all over the cell. These essentially start pinching the cell into two parts. So essentially a balloon being pinched in the middle, you will form two separate balloons that pinch the cytoplasm from the middle into two separate cells. This occurred in the animal cell. But in animal cell, we know that there is no rigid cell wall present, there is only cell membrane, and this can occur easily. But in case of plant cell, there is a rigid cell wall present that cannot be pinched so easily by cytoskeleton elements. So in plant cells, what happens is, on the other hand, it consists of a stiff cell wall that prevents such contractile cytokinesis or cytokinesis by cytoskeleton contracting. So instead, what happens is that a cell plate forms. So see, this is a new thing that is forming in the plant cell only, which is known as cell plate, which essentially divides the cell into two parts. So this essentially you're putting a cell wall in middle of the entire cytoplasm. So a cell plate structure forms that divide the cytoplasm into two separate cells by forming a new cell wall in the middle of the cytoplasm. So essentially, since the cells cannot physically divide into two, we are erecting a physical barrier in the middle of the cell, which is the cell plate, which will later develop into the full-fledged cell wall, which separates the plant cell into two. So this is how cytokinesis occurs in both the animal cell and the plant cell. So this was the entire process of how exactly cell division occurs. I hope that is fine. Okay, so we move forward to answer our first question. That is, the cell cycle is a set of activities that take place inside cells to produce daughter cells by dividing the cell cytoplasm and nucleus, as we just saw. The cell cycle has four key stages, that a cell goes through. In any eukaryotes, what is the ideal stage for counting the total number of chromosomes? And also, at what point do chromosomes align on the equator of the spindle fibers? So, as we have just discussed for the second part only, what point do chromosomes align on the equator of the spindle fiber? We have seen that metaphase is the phase at which they align at the metaphase plate or the equator of the cell. And as I said, that in this phase only, the chromosomes are the most condensed and occur in a very like a straight line or in a way same plane, which allows very easy counting of the chromosomes as well. So, answer to the question like what is the ideal stage for counting the total number of chromosomes, that is metaphase. And at what point do chromosomes align on the equator of the spindle fibers, that is also metaphase. So our correct option is option B. So that was that question. Anyone wants to ask anything from this?
So, you have said that begins chromosome in the metaphase clearly. But when the cell is not in the growing phase or it is not in the metaphase, it is in normal condition, can we see the chromosome
Yes, we can see the chromosome. The thing is that
genetic material?
so what the chromosome looks like is something that right here. So you can see that the chromosome is stretched out to form a string-like structure, and this is how the chromosome exists during normalcy. So no cell division occurring, but it is present very compactly in the nucleus. Further, the since the strings can be close to each other, and since here it is colored as blue and red, but in the normal cells you will just look at whatever dye is staining the chromosome. So everything will essentially occur orange. So consider a number of strings in this location only that are flowing over each other, and everything is colored orange. So you essentially can't say "Okay, that is the chromosome number 1, that is chromosome number 2, that is chromosome number 3" very easily. That is one point. The second point is that these studies have been extensively carried out during metaphase and other cell division phases where the chromosomes are present in the form of sister chromatids. And these have been extensively identified because the X-shaped structure that we see right here uh, is very defined for each chromosome. So the shape and the structure of the chromosome number 1 in this cystochromatid form will be very different from chromosome number 20. And this can be easily identified using various dyes, and that can tell us which chromosome it is and where it is present. So here only looking at this structure we can say this is say chromosome number 1, this is 2, this is 3, this is 4. Easily count the number of chromosomes and then report it as is. So these are the two reasons for which like counting is not done during interphase. So because two decondensed chromosomes not easy to count, and because in metaphase and sister chromatic form they have been well identified and characterized, so that one look at it we can say which chromosome it is out of the 23 that represent in humans, or in case of E. coli whatever the number is. Is that fine?
Yes, sir.
Okay. So that was that. If there is no other question, then we can move forward to the next one. So the next question is: assume you are studying the cell division in a metastatic cancer cell line. Your senior has given you a subline of cells where she has fused the kinetochore proteins with GFP. So GFP is the green fluorescent protein which fluoresces green in color. A protein then that produces green fluorescence. After reaching confluency, that is your entire cell plate has is full of cells, like you have cells everywhere on your plate. You harvest the cells and mount it on a slide. Essentially you take the cells and you put it on a slide to look at it under the microscope. Which part of the cells do you think will show fluorescence? If you harvest the cell at the logarithmic growth phase followed by cell lysis and centrifugation, then what part of the solution will contain the most green fluorescence? So after looking at the cells under the microscope, we went ahead and harvested the cells. So essentially, you take up the cells from the solution, from the plate, the solid plate, and put it into a medium, a liquid medium. You lyse the cells, so break down the cell membrane, and then centrifuge it. And then, what do you think? Where does the green fluorescence will occur? So these are the options that we have. For the first part, which part of the cell do you think will show fluorescence? We have both chromosomal ends or telomeres will show fluorescence. And when you centrifuge the cell lysate, you will see the supernatant will contain the majority of the green fluorescence. The second option is throughout the entire length of the chromatids is where you will see fluorescence. So the entire chromatid will light up with fluorescence, and the pellet will contain the majority of the green fluorescence. Third option is the centromere only will show fluorescence, and when you centrifuge a cell lysate, you will see that the cell pellet only contains the fluorescence. And finally, B and C, so these two options can both be correct. So GFP fused to kinetochore. First, you want to look at the cell under the microscope and see where they are present on the chromosome itself. And second, you lyse the cell, take out the cell lysate, centrifuge it, and try to find in which part the. So when you centrifuge a cell lysate, first you will have two parts or in the particular tube. One would be the cell pellet that contains every cell organelles, the nucleus, the genetic material, various large proteins that are attached to the cell pellet present at the bottom of the tube. And on the top of the tube, we will have a liquid that is known as the supernatant. These contain mainly various smaller proteins and organelles that very smaller proteins and plasmids that are escape out of the cell and reach into the supernatant and dissolve there. So undissolved material cell pellet, dissolved material in the supernatant itself. So the question is that where will the fluorescence occur? The supernatant or the pellet? So those options are given here. Supernatant will contain fluorescence, pellet will contain the fluorescence, pellet will contain the fluorescence, and for B and C, both of them could be correct. So I would encourage everyone to try and answer this, like put your whatever you think is the correct answer in the chat box. A, B, C, D.
Let's see options. Centromere only pellet will contain most of the fluorescence.
Okay, Rishita. So Rishita says it is C, centromere only. The pellet will contain most of the fluorescence. Shreya also says the same, that the pellet will contain most of the fluorescence. So let us go forward and try and understand why this occurs. So first we have to a brief discussion on kinetochores. As I just said, like just discussed, that kinetochores are large protein assemblies that connect chromosomes to microtubules of the mitotic and meiotic spindles. So mitotic spindles and meiotic spindles are the same as we just discussed. They connect to the chromosomes and help the division of the chromatids. And in order to distribute the replicated genome from a mother cell to its daughter cells. It is a disc-shaped protein structure associated with duplicated chromatids in eukaryotic cells, where the spindle fibers attach during the cell division to pull the sister chromatids apart. So essentially, this is the connecting point between the microtubule and the particular sister chromatid. And once the pulling of the once during anaphase the microtubules contract, they will pull one sister chromatid with each other. So this is what the entire concept around kinetochores is. So the kinetochores, another important thing is that the kinetochores is a protein complex that attached near the centromere. So centromere is the middle of the chromosome, which is present right here, which divides it into two these dumbbell shapes. So that is where the kinetochore is present. So with the entire information from that particular slide, we know that if GFP protein is fused to the kinetochore, then only the centromere should show us fluorescence and nothing else should show us fluorescence. And since after centrifugation, the genetic material, the DNA, the chromosome, stay with the cell inside the cell pellet, trapped inside the cell, then That is why only the pellet should contain most of the green fluorescence and nothing else. The supernatant shouldn't contain any of the fluorescence. So that was the second question. Anyone wants to ask anything from this?
Mhm.
Okay. So we move forward to the third question. The diagram shows a series of events that happen during the cell cycle in eukaryotes. Arrange this in the correct order. What is the maximum number of times the chromosomes can replicate per cell cycle in human keratinocytes? This is the second question. What is the maximum number of times the chromosome can replicate per cell cycle in human keratinocytes? So this is essentially a jumbled up form of the entire cell cycle that is occurring. And we are now asked to arrange this. So which step occurs after which? So the four options give various orders of the cell structure. So various orders of these structures being formed, and we have to choose which is the correct option. And maximum number of DNA replication is also given as part of the second question. So I would encourage everyone to try and think this through and answer this. Which do you think is the correct order that occurs during cell division? So you can post your answer right into the comments. Think this through. What do you think after our entire discussion is the order of cell division, and how does the nucleus look like it during cell division?
D option.
So Vishita says that the correct option is D, which is 2 followed by 3 followed by 4 followed by 1 followed by 5. Okay. Shreya says the correct option is A, that is 3 followed by 2 followed by 5 followed by 4 followed by 1. Okay. So if there are no more options, then we will try and answer this particular question. So we will go back to our previous slide on mitosis itself. So going through mitosis, we have interphase, in which first we have condensed chromatids forming condensed chromatids that are formed inside the nuclear envelope, followed by prophase, where we have sister chromatids forming. So the X type of structure from a one dot type of structure. Then we have alignment of the chromosomes at the metaphase plate, separation of chromosome at the anaphase, so pulling apart. Finally, telophase and cytokinesis, that gives us again the identical starting point, nuclear membrane that encapsulates the chromatids uh, like this and into identical daughter cells. So looking at this particular mitosis picture, if we go back here, we can see uh, in the starting, that is during interphase, is where we have one centrosome and the entire chromosome is in the string kind of structure formation like here, and we have nuclear membrane that is intact. Next, what happens is that the centrosome replicates into two, and the chromosome thickens and starts to condense. So 3 followed by 2. Next, what happens is that the string-shaped chromosomes condense even further to form the X-shaped sister chromatids. Right here, we have still formation of the presence of two centrosomes, and the nuclear membrane finally collapses. Then they are arranged in the middle of the cell in the metaphase plate, where they line up essentially in one line, and finally in the first one where they are pulled apart towards two different poles. So this is interphase. This is still early prophase. This is prophase. This is metaphase, and this is anaphase. So the order goes 3, 2, 5, 4, and So Shreya answered correctly that it is option number 1. 3, 2, 5, 4, 1 is the order. And the maximum number of times the chromosomes can replicate per cell cycle is same for every normal cell, irrelevant if it is a keratinocyte or a myocardiocyte. It will still remain number of times the chromosome can replicate in a cell cycle to be 1. So the correct option is number 1. Is that fine? Any questions from this?
Can you hear the second question asked in this number of time chromosome can replicate?
So the number of times chromosomes are replicating. So as you can see, during the interphase we have one chromosome right here and second chromosome right here. So when it goes to the prophase, this chromosome divides into two to form sister chromatids. So one deep blue rod-shaped chromosome divides into two deep blue rod-shaped chromosomes that are attached in the middle to form an X type of structure. So essentially, one chromosome formed another. So there is one duplication or one replication, one step of replication. So once it is replicated. Similarly, the light blue one, which was rod shaped, forms an X shape. So one round of replication for the light blue one as well. So the formation of light blue to two light blues. So essentially, one round of replication that is occurring. So in all normal cells, only one round of replication occurs from interphase to prophase. So that is what is being discussed. Maximum number of DNA replication. The DNA during cell division, mitosis especially, divides only once in order to form the daughter copies. In case of normal somatic cells that are present in our body. Okay, so we will move forward to the next question. So the next question is, in a cell culture laboratory, you are looking at a glass slide that exhibits a certain phase of cell division. When you questioned your senior about the right phase, he said it was anaphase. You attempted to match the slide to some of anaphase's important crucial points which show which demonstrates that it is actually anaphase. So choose the correct statement about anaphase. The only one option out of the four is correct. The first one is, as chromosomal decondensation occurs, sister chromatids separate from one another. Chromosomal reformation happens just after completion of crossing over at this stage. Homologous chromosomes segregate from each other, moving towards pole because of microtubule depolymerization, which happens at the kinetochore, and depolymerization of the polar spindle fibers causes sister chromatids to stop segregating at this point. So, knowing what we know about anaphase, which of the following four options do you think demonstrates anaphase or signifies anaphase most correctly? You can go ahead and put your what do you think is the correct option in the chat box.
The option.
Okay. So Rishita says it is option A. As chromosomal decondensation occurs, sister chromatids separate from one another. And Shreya says it is option C. Homologous chromosomes segregate from each other, moving towards pole because of microtubule depolymerization that happens at kinetochore. Okay, so if anyone else wants to try and answer this, please go ahead.
Let us see differentiation.
Okay, so Rishita changes her answer to C. So anyone else that wants to answer? Okay. So let's go forward and try and answer this. So the option that both the Rishita and Shreya pointed out, C, anaphase is the correct one. C is the correct one, and let us now look at closely at anaphase and understand why. So as we have seen before, that in anaphase, each sister chromatid is pulled apart by kinetochores that are attached to the poles. And what happens during this is that the protein complex holding the sister chromatids breaks down, forming individual chromosomes, and each pair of the chromosome is pulled to the poles of the cell. And microtubules that are not normally attached to the chromosomes, known as normal microtubules, and the ones attached to the chromosomes are known as kinetochore microtubules. They elongate and push the poles apart, giving the cell an elongated morphology. So essentially, the main point during anaphase is that the sister chromatids separate from each other and are now pulled to either pole. So knowing just this information about anaphase, we can go ahead and choose the correct option, that the homologous chromosomes segregate from each other, moving towards pole because of microtubule depolymerization that happens at the kinetochore. So microtubule depolymerization is the essentially the process via which the microtubules contract. But that is not very important to us because segregation of homologous chromosomes is the main point that we have to look at. So homologous chromosomes segregate during anaphase, and thus option C is correct. Any questions from this particular slide? Okay. So we move forward to the next question. So this is also an interesting question. The graphic below depicts the sequence of events that occur throughout the development of an embryo. So now we are changing track and moving from cell cycle and cell cycle regulation to developmental biology. So the graphic below depicts the sequence of events that occur throughout the development of an embryo. Select the appropriate answer for 1, 2, 3, and 4 from the below mentioned options. So essentially we have to choose which stage during development are these. So these are the images, and 1, 2, 3, 4 denote the various processes that are occurring. So we have 4 options, only one of which is correct. The given options include fertilization, blastulation, gastrulation, cleavage, and organogenesis. So you can go ahead and try and answer out of A, B, C, D, which of the options match the correct stages that are given right here. Shreya answered that the correct option is D, fertilization followed by cleavage followed by blastulation, blastulation followed by gastrulation. Okay. Okay, so we will go forward and try to understand what is the correct order via the following solution slide. So we have a video right here. You have to tell me if the video is playing with sound or not. Okay.
We're gonna.
Okay. So you have to tell me if the sound is playing or not.
We're gonna talk about early embryogenesis. Say you're an egg cell.
So is the sound playing? You can hear the sound over the meet. Yeah.
Yes, sir.
Okay. So I have also turned outside the subtitles for your help. So we can go ahead and look at the video.
We're gonna talk about early embryogenesis. Say you're an egg cell, and you have this nice thick outer glycoprotein coat called the zona pellucida, and you've got your plasma membrane just inside of that. And a sperm has made its way through the zona pellucida and managed to get in through your plasma membrane and merged its genetic material with yours. You're now called a zygote, and you'd like to go on to form an embryo, but not much is going to happen when you're stuck as a single cell. And so what you've got to do is divide into multiple cells, and you've got to do it fast. In fact, you've got to do it so fast that you don't have time to grow. So you actually just split into 2 cells. And this process of splitting without growth is called cleavage. And you do this a number of times, dividing from 2 cells to 4, and from 4 cells to 8, all the while staying within the zona pellucida. So you've gone from 16 cells to 32 cells, and at this point you look different enough that somebody decides to give you a new name. So instead of being called a zygote, you're called a morula. And morula is just a Greek word that means mulberry, and you actually do look a little bit like a mulberry. So here's a picture of a mulberry from my front yard to prove it. This one's not quite ripe. So you're finished with the cleavage stage of early embryogenesis. You've gone from 1 cell and just divided without growing at all into 2, 4, 8, 16, and then 32 cells. And now more interesting things start to happen. So you're still stuck within that zona pellucida. So we'll draw it in here. And I'm not going to keep track of the number of cells anymore, I'm just going to draw them in. But what you'll notice is I'm drawing them in quite compacted. I'm drawing them all right in the middle of the structure here. And they do look like they're a little bit tighter together. And this is a process that's called compaction. The different cells within the morula start to get closer and closer together. And in fact, the cells start to get a little bit different from each other too. And you notice that these cells on the outside are a little bit different. I'll draw them in here in a slightly different color. And we can tell them apart, and this process of being able to tell cells apart as they become different things is called differentiation. So here we have 2 separate populations of cells. This one on the outside, we'll call them trophoblasts, and this mass of cells on the inside, we'll call embryoblasts. And so you're going to continue with your process. We'll move on to the next stage. Again, we'll draw our zona pellucida here, and we'll draw all of our trophoblasts along the outside here. Another interesting thing is happening to those cells in the middle. They're starting to clump even more. In fact, they clump so much that they all cluster at one end, leaving a little cavity on the other end. So here you can see your trophoblasts and your embryoblasts, this mass of cells in the middle. Some people actually call that the inner cell mass, and it's left you with this cavity, and that cavity is called a blastocele. Now, again, you are starting to look a little bit more different than you have in the past, so we're going to give you a new name. This structure that you've turned into with a blastocele and an outer cell ring of trophoblasts is called a blastocyst. In fact, we name this whole process after you, and after cleavage, the process is called blastulation. So also about this stage in blastulation, your zona pellucida starts to disintegrate away. Here I'm going to draw little bites being taken out of it here as it falls away and disintegrates. And that's going to be important later on because you can't be stuck in this thing forever. And the next step in blastulation, you actually just lose it completely. So now we'll draw our trophoblasts completely naked without a zona pellucida. But more interesting things are happening to you. Your inner cell mass of embryoblasts is starting to look a little bit different. So you still have this rim on the outside here up at the end of your blastocyst, and you still have this mass here in the middle. But you've developed another cavity, and this cavity is called the amniotic cavity. And also, your inner cell mass of embryoblasts has started to differentiate more. And now it's got this layer on the bottom of it here, and the cells in this layer are called hypoblasts, while the cells in the layer just above it are called epiblasts. Now at this stage, we're pretty much completely free of our zona pellucida, and that's going to be really important for implantation. But that's a discussion for another time. We're going to focus on this ball of cells here. And I really want to stress that this is a ball of cells. It's not flat like we've got it drawn here on the computer screen. It's three-dimensional. It's spherical in nature. A spherical melon. So think of it like a melon. Here I'm going to draw a spherical melon. So now I want you to picture taking a big knife or a machete or something and just slicing the top off the melon like this. Here we'll erase the top part of our melon here, and we're left with a flat surface. So now that you've got a melon with a flat surface, picture yourself taking a pancake and just putting it right on top of this flat surface like that. And that's basically what we've got here with our blastocyst and this forming layer of epiblasts and hypoblasts. It's really a pancake. It's not two-dimensional here. It's a three-dimensional disc of tissue. So we're going to draw this again, but this time without the outer sphere of trophoblasts. But we're going to draw our pancake of epiblasts and hypoblasts. And what you can see here is our pancake has got 2 layers. So in fact, this is a very important structure in embryology called the bilaminar disc. And we're going to get another look at the bilaminar disc here. So I'll show you. We're actually going to draw a little plane through the disc here. And if we take a cut view through this plane that I've drawn, it looks a little bit like this. So this is still our bilaminar disc, but now we're looking at a slice through the bilaminar disc, as opposed to here we're looking at the entire bilaminar disc from the outside. So now we're looking at our pancake of our bilaminar disc here, and we notice we start to see something forming on the edge of it here. And that's something that looks like a little bit of streakiness that kind of splits our pancake right in two. You can think of it as pouring a little streak of syrup along the top of the pancake and dividing it into 2 halves. So here I'm going to draw it on the surface of the pancake, and then over here on our cut view, you can see that this little streak happens right about here in the middle as it reaches the plane of our image. And this streak of syrup on our pancake has got a name. It's actually called the primitive streak. And the formation of the primitive streak marks the beginning of the next stage of early embryogenesis, and that's called gastrulation. Now what that primitive streak actually is just the site where the cells in this epiblast layer of our bilaminar disc start to migrate. Here I'll draw the paths of the migrating cells. They heap themselves up right at the primitive streak, and then they start burrowing their way down to this bilaminar disc. And they go out into the hypoblast layer, and they just kind of go all over the place here. But it all happens from that primitive streak. So as the cells migrate out from the primitive streak, we can see that our structure has changed a little bit. Now instead of having 2 layers, 1 layer of epiblasts and 1 layer of hypoblasts, all of those migrating cells have now differentiated even further, and we're left with a layer of cells on the top, a middle layer of cells as they go out into the body of the bilaminar disc, and we still have a lower layer of cells down here. And so now you can see, instead of the bilaminar disc that we actually have a trilaminar disc. So we have 3 layers, 1, 2, and 3, and these are actually called our germ layers. So we have a single layer on top, and instead of epiblasts, this layer is now known as ectoderm, and our middle layer is mesoderm, and our lower layer is endoderm. And cells from these 3 layers go off to do very important things, and each layer forms its own specific structures. So that process of the formation of the 3 layers is called gastrulation. Once our 3 layers are formed, we can move on to the next step in embryogenesis, and the next step is called neurulation. So we'll draw our 3 germ layers in. We have a layer of ectoderm on the top. In the middle, we have a layer of mesoderm. And then on the bottom, we have our endoderm. So this final stage in early embryogenesis is called neurulation. And as you might expect with neuro in the name, we're going to see some neural elements formed here. And now we have our trilaminar disc with our 3 germ layers. And in the middle of the mesoderm, the central layer here, we start to have further differentiation of cells. And right in the middle, we get this cord that starts forming. And I'm going to draw it as a little purple dot here, but what that really is underneath where the primitive streak was, mesoderm cells are differentiating into a cord structure, and it's called a notochord. Now in humans, the notochord doesn't go on to do a whole lot. It does form part of the intervertebral discs, and very rarely it will cause a tumor called a chordoma. But in general, its main goal and its main purpose is here in neurulation. Now, this differentiated bit of mesoderm actually induces a change in the ectoderm above it. So here we get a change within the ectoderm. I'll draw that in as kind of a thickening of the ectoderm right here. And that thickening in the ectoderm has a name, since it's kind of plate-like when we look at it in section here. This is called the neural plate. Now we have our notochord formed and we have our neural plate formed. The next thing that happens, so we'll redraw our 3 layers here, our ectoderm, our mesoderm, and our endoderm. And here at the bottom we have our notochord forming within our mesoderm. And right above it, those neural plate cells actually start to dive into the mesoderm. And as those neural plate cells dive in, they start to form a ring structure. And actually, since this is a 3-dimensional thing, this ring is more like a tube, so you can picture this tube going off into the rest of this pancake that we've drawn here. And as this neural plate zips up and dives down into the mesoderm, it becomes known as the neural tube. Now, this isn't a perfect process, and as the neural tube is zipping up from one end of our pancake to the other, little cells are breaking off from the ectoderm and going out into the mesoderm. Now, these cells actually have a very important role as well, and will go off and to differentiate into their own special tissues, and these are called neural crest cells. So once we have the formation of our neural tube and our neural crest cells are diving off into the tissues and differentiating, and we have a pretty good idea of what the ectoderm, mesoderm, and endoderm are, our early embryogenesis is complete. So just to recap, we started off as a zygote, a single cell that was fertilized by a sperm. Cleavage happened, we didn't grow, but we split into a whole bunch of cells and developed into a morula. The morula cells started to differentiate, and we developed trophoblasts on the outside and embryoblasts in the middle. We formed a little cavity and became known as a blastocyst. After the blastocyst cavity formed, or the blastocele, a second cavity called the amniotic cavity formed, and a pancake of cells across the sphere became apparent. Those cells continued to differentiate, and we formed a primitive streak. The epiblast cells dove into the primitive streak and started to differentiate into our three layers that became our ectoderm, mesoderm, and endoderm. And in the final stage of neurulation, we had development of a notochord, which induced a neural plate to form, and the neural plate dove into the mesoderm and formed the neural tube. Neural crest cells associated with the neural plate also went off into the tissues to further differentiate.
So that was the entire video. So just coming back a bit,
in a pink form,
here we can see the order of processes that occur. First step is fertilization, followed by cleavage, followed by blastulation, followed by gastrulation, and then neurulation. So this particular video delved deeply into early embryogenesis and covered essentially entire thing very briefly, but gave us enough information to answer our particular questions that we have been asked and clear up various concepts regarding earlier embryogenesis.
We developed.
So yeah. So in this particular image, you can see that the coming back to a particular question, when we see the various steps of our schematic, the first step in which we have an egg being fertilized by a sperm, this first step is fertilization, followed by cleavage of the cell into two cells without division, a division into two cells without growing, which is known as cleavage, followed by division into a blastocyst stage, and finally formation of the three germ layers, which is known as gastrulation. So the 1, 2, 3, 4 involved fertilization, cleavage, blastulation, and finally gastrulation. That is option D is the correct option. So any questions from this particular slide?
No.
Okay, so we move on to the next question. The question is, rapidly proliferating cells may begin to appear aberrant and disordered as they continue to undergo genetic change or mutation. This stage of a transforming cell can be described best by which of the following options? On the other hand, uncontrolled cell division may result in overabundance of cells that appear normal under a microscope. What do you call these conditions? So once the first option, aberrant or disordered division of cells which are apparent under the microscope, and high rate of cell division which is not very apparent and appears normal under the microscope. What are these two called? So we have four options. First is metamorphosis and dysplasia. Second is hyperplasia and hyperproliferation. Third is dysplasia and hyperproliferation. Fourth is hyperextension and hyperplasia. So whatever you think is the correct option, you can go ahead and put it into the chat box.
No, I think so.
Okay, no problem at all. So let us go and look at the solution slide and try and understand this. So first let us understand some terms. First term we want to know about is dysplasia. Dysplasia, so dysplasia from the name, dys, dys means bad, and plasia means form or shape. So essentially, the entire term means bad shape or bad form. Dysplasia is the term that is used to describe the presence of abnormal cells within a tissue or an organ. Dysplasia is not cancer, but it sometimes may become cancer or give rise to cancer. It has several severity stages: mild, moderate, severe, depending on how abnormal the cells look under a microscope and how much of the tissue or the organ is affected. Normal cells may also go on to become, these normal dysplasia, normal cells can go on to become cancer cells as well. Before cancer cells form in tissues of the body, the cells go through abnormal changes called dysplasia. In dysplasia, the cells look abnormal under the microscope, but these cells are not yet cancer. These just look abnormal because high rate of cell proliferation, and this can be looked and separated out based on what you see under the microscope. Another very similar process is known as hyperproliferation. Hyperproliferation, on the other hand, is the rapid growth of cells that may occur while normal development as well. So during development, we know that we rise like grow from a single cell to a multicellular organism containing various organs. So it is obvious that we need a huge amount of cell growth to occur between those two time points. So this occurs by the process known as hyperproliferation. We see a huge increase in cell numbers, huge cell proliferation, but still, when you see the cells under a microscope, they do not look very abnormal, but they look normal, just they are dividing very fast. In this case, the cell density of the tissue, that is the number of cells per unit volume, may increase, but the growth appears normal morphologically, and cells still show contact inhibition. So contact inhibition is something that we touched upon, is when cells, when they collide with other cells or come in contact with other cells around them, they do not proliferate any longer because they understand that there is no space anymore or enough nutrients to continue proliferating. So these rules are followed by hyperproliferating cells. This is essentially the main difference between both of them. Dysplasia cells look abnormal under the microscope, may or may not show contact inhibition. Hyperproliferating cells look normal under the microscope and show every morphogenic marker or cell contact inhibition to show that they are actually normal cells just dividing very fast. So this is a schematic showing the process in which normal cells may become cancer cells. So first we have normal cells that look like this. Then we have hyperplasia or hyperproliferation of cells, which we see a huge increase in cell number, but this is still normal that we can see. Then we have dysplasia, in which the cells essentially look abnormal, as you can see right here. Nucleus is looking strange. Cells essentially bump up want to grow on top of each other. This is dysplasia. Finally, we have cancer, in which we have abnormal growths that occur into the tissue. So this is the difference between dysplasia and hyperproliferation. So coming back to our question, we have rapidly proliferating cells may begin to appear aberrant and disordered as they continue to undergo change or genetic mutation. This stage of a transforming cell can be described best by which of the following options. Uncontrolled cell growth or division, on the other hand, may result in an overabundance of cells that appear normal under the microscope. What do you call these conditions? So, our transforming of cells by genetic change that they look abnormal or aberrant is dysplasia, and uncontrolled cell growth or huge amount of growth of cells to form a large number of other cells but they appear normal is hyperproliferation. So the correct option is C. So any question regarding this?
In hyperproliferation, this cell appears to seem normal under microscope.
Yeah.
So, are there any side effects or kind of medical issue due to hyperproliferation?
Okay, so hyperproliferation, as I said, is something that is only seen while development, or where in case where regeneration or high speed regeneration of tissues is seen. So this does not occur in the adult body, or your somatic cells will not undergo hyperproliferation ever. So that is a very bad thing that occurs during adulthood. So such a thing will not occur, and thus there is no question of this hampering any adult state, causing a disease. So if hyperproliferation does occur during adulthood, this will just go and give rise to dysplasia, which might give rise to abnormal growths like tumors in the body. So that is a disease in and of itself, but not hyperproliferation alone is not a disease and does not occur in adults, but it can occur, and that will be part of the dysplasia growth which will occur in further stages, which may give rise to tumor like growth and can grow into a cancer. Is that fine?
Yes, sir.
Okay, so we move on to the following question. What type of cell combinations? So the next question is on animal cloning. So this is something that was discussed in the particular course. So you can go ahead and let us go ahead and tackle this particular question. What type of cell combinations should you start with if you wish to clone an animal, for example, a lab rat? Select the right response out of the following four. The options are early embryonic stem cell for donor nucleus and female rat eggs for recipient, any rat cell for donor nucleus, female rat embryonic stem cells as recipient, only male somatic cells as donor, and embryonic stem cell nuclei as recipient. Any gametic cell can be used as a donor, and any adult somatic cell can be used as a recipient. So only one of the four is correct. So this is only one option type of correct answer. So please go ahead and try and answer this. So what do you think is the combination needed for cloning an animal? You can put here whatever you think is the correct answer in the chat box.
First option.
Okay. So Shita says that it is option A, or the first option, that the early embryonic stem cell for donor nucleus and female rat eggs as recipient. Any other answers are forthcoming? We can go into the solution. Okay, Shreya also says that option A is correct. Early embryonic stem cell for donor nucleus and cell rat eggs for recipient. Okay, so let us try and answer this in our solution slide. So the process that so cell cloning can be done via 2-3 ways. The process that we are talking about here is somatic cell nuclear transfer, SCNT. SCNT essentially, or somatic cell nuclear transfer, requires two major steps. The first is a somatic cell, as the name suggests, from which you will take a nucleus. So you will take the nucleus out of a somatic cell, or which is essentially a normal body cell. The second important factor is an egg cell. So that is normally fertilized using a sperm, but in case of egg cells, normally is fertilized with a sperm, but this has not yet been fertilized, and it is a haploid cell. What you do is that you remove the nucleus from the egg cell and you fuse the somatic cell nucleus with the egg cell. So, an important thing to understand here is that the egg cell was haploid, or had only one set of chromosomes, and the somatic cell is diploid, or has two set of chromosomes. So, essentially, it looks like as if you have artificially infertilized Uh, you have artificially fertilized the particular egg cell because you have made the egg cell go from the haploid status or one set of chromosomes to two sets of chromosomes right here by replacing the nucleus with the somatic body cells nucleus. And now if you let this particular egg cell grow, it will grow and divide like a normal embryo does and produce a number of clones. So this particular clone of cells can take two paths. The first is reproductive cloning, in which you plant this particular set of cells into a surrogate mother and allow this to grow into an embryo and a new organism. So that is known as reproductive cloning. And next is therapeutic cloning, in which you look at the particular cells, allow them to proliferate to a particular stage, and then segregate them out, culture them in a tissue, culture them in a tissue culture facility, and grow them as you need into various organs or tissue types that you need that can be directly used for therapeutic purposes. Because these are egg cells, so because at this stage it is still an embryonic stem cell. So at embryonic stem cell, what it does is that it is pluripotent in nature, that it can form any tissues in our body. So using these particular cells to grow various tissues, say you have need but a specific tissue that is lacking, you can grow it using therapeutic cloning, and then go ahead and transplant into a particular patient and treat them of their disease. So these are the two type of cloning that can be done: reproductive cloning and therapeutic cloning. So essentially, SCNT requires two major steps. One is a body cell which has a diploid nucleus and an egg cell whose nucleus is removed. These are the only two steps that are required. Is this fine? Okay. So now we go to this beautiful picture in which we'll understand how exactly this process was used in cloning Dolly the sheep, which is the first cloned animal ever. What was done is that first we took an adult fin dorset ewe, that is a adult female sheep, and took its mammary gland cells, which will be its somatic cells. And as the process was, we so what we do is that we arrested its cell cycle and essentially kept its nucleus. And from the other end, we have a Scottish blackface female sheep. We take its unfertilized egg cell, as we had to take out take out its nucleus. So we enucleated the egg cell and then fused it to our donor cell. So we have essentially artificial fertilization present here, nucleus from the somatic cell and the cytoplasm from the enucleated egg cell. And we allowed it to divide to a particular stage and then implanted into a surrogate mother and allowed it to grow. So essentially reproductive cloning is done here, and this embryo will now grow to give rise to a new organism, which is the fin dorset lamb Dolly. The important thing here is that the Dolly is essentially a fin dorset lamb. That is, its genetic makeup is the same as the donor nuclei's genetic makeup. That is, it is not a blackface, Scottish blackface, as the surrogate mother, or the from the one we have taken the egg cell from, but the new organism will have the genetic makeup directed by the nucleus donor itself. So we have Dolly, who is a fin dorset lamb. So this is how cloning of Dolly the sheep was done, and reproductive cloning in a nutshell can be done. So any questions from this particular slide? Okay, so I hope this was understood. So we go forward to answer our particular question. What type of cell combinations should you start with if you wish to clone an animal, for example, a lab rat? Select the right response. The correct option is A, as both of Shreya and Rishita has pointed out, that you have to take an early embryonic stem cell for donor nuclei, that is a somatic cell, which a diploid cell, from which you will take a diploid nucleus, and a female rat egg, which can be then enucleated and used as the recipient. So this is essentially the combination that will work. Yes, Rishita, do you have anything to add?
Yes, sir. Sir, can change the gender, like can we take the nuclei from the female egg and um can we take the sperm instead of egg? That is
If we take the sperm
nucleated.
instead of the egg in this but enucleated cell. So the thing is that the sperm only contains very little genetic information. By the sperm contains enough genetic information, but it does not have the machinery to form the entire embryo. As you know, that the sperm cell is very small and only contains the genetic information only. It does not have the cellular machinery to go on to divide and form the entire embryo. That is only present in the egg. So that is why only the egg can be taken in this case to form to do this experiment, because it has the cellular machinery to go ahead and do the entire thing.
Okay, so we can only clone the male organism and not the female because
Yes, yeah, you are correct. So we have to start with the female organism for the egg donor, because only that way we can go ahead and clone it in this particular thing. So that was this particular question. Now we move forward to the next one. So in laboratory, you are preparing for a cloning experiment. That will necessitate the use of embryonic stem cells. Choose the correct combination of embryonic stem cell phases. So the options are given here. So these are information about the embryonic stem cells, and you have to choose which one of them are correct. The first option is they are unsuitable for cloning studies. They can be easily extracted from the blastula stage. These cells can multiply endlessly in specific culture media, and they are highly specialized cells. So you have to choose the correct option, which only show the correct combination of these 4 points. So you have 2 and 3, 1, 3 and 2 and 3, 2, 3 and 4, and 1, 3 and 4. So Shreya answers that the correct option is A, that is 2 and 3. That is, they are easily extracted from the blastula stage, and these cells can multiply endlessly in the specific culture media. Okay, so any other answers forthcoming, or we can go ahead and answer this particular question.
no idea.
Okay, so information about the embryonic stem cells. So yeah, embryonic stem cells are shown right here. So as we have seen the entire process of early embryogenesis, embryonic stem cells are present in the blastocyst stage in the inner cell mass. So these cells are essentially the embryonic stem cells. So if we extract these inner cells, they can be plated on a media on a plate, and can be allowed to grow into various types of specific types of cells that we need in the adult. So these embryonic stem cells are present in the inner mass of cells at the blastocyst stage of the embryo, and this can be easily extracted in this particular stage. They are pluripotent in nature, which is a very important point. That is, they can give rise to all cell types, or the all embryonic cell types, and by and obviously it can also give rise to every kind of cell type that we have in an adult body as well. They can be immortalized and allowed to grow indefinitely on a plate. They can be extracted from developing blastocysts and are regularly used for cloning, transgenics, and knockout creation. So they are extensively used in cloning studies as well. So this is an image showing that how what the pluripotent stem cells can do. They are taken out from the blastocyst, and they can go ahead and form various differentiated cell types, including red blood cells, fat cells, cardiac muscles, enterocytes, chondrocytes, epithelial cells, and neurons. It can form anything depending on the culture media that is given to it and various cues supplied. So, embryonic stem cells are present in the inner cell mass of the blastocyst, are pluripotent in nature, can grow indefinitely, and are regularly used in cloning. Taking all of this together, we will go ahead and try and answer this question. So, the first option, they are embryonic stem cells are unsuitable for cloning studies. That is false. They can be easily extracted from the blastula stage. That is true. These cells can multiply endlessly in specific culture media. That is true. And they are highly specialized cells. That is false. Stem cells are completely undifferentiated or unspecialized. They can give rise to a number of specialized or differentiated cells. So, depending on this particular question, the correct option are 2 and 3, and that is given in option A. That is why option is A is correct. So, Shreya, you were correct in choosing option A. Any question from this particular slide?
No, sir.
Okay. So, let us move forward to the next question. So, we are now looking at cellular instability and formation of cancers. Tumors are assumed to be benign in their early stages until a small number of cells escape the tumor origin and over time develop a secondary tumor at various sites organs of a body via a highly specialized process. So, a tumor is present at location A, it can escape from that particular location and spread to other parts of the body and begin a secondary tumor at that site. This is via a highly specialized process, and we are asked what exactly the name of the process is, and also figure out what attribute does not constitute a malignant tumor. So, which of the following is not a property of a malignant tumor and can occur normally as well in other cells? So, we have the four options here. The options are A option is translocation and angiogenesis. So, the process of mm, going to another location is translocation, and the attribute is angiogenesis. The process is neoplasia, and the next is invasiveness. The process is translocation. The attribute is genomic instability, and the process is metastasis, and the attribute is pluripotence. So only one of the following options is correct. You can go ahead and try and answer this. Put the answer in the chat box if you think you know it. Okay, so Rishita says that it is option A. Shreya says that the option is option D. Okay, so let us go ahead and try and answer this particular question. So the first option, so the first question, that what is the process via which the cancer cells move from one side to the other, is known as metastasis. So let us get to know a bit about metastasis. So metastasis is a spread of cancer cells from the place where they first formed to another part of the body. In metastasis, cancer cells break away from the original primary tumor, travel through the blood or lymph system, and form a new tumor in other organs and tissues of the body. So, this is the process via which they escape from the primary location and end up in a second location. Metastasis occurs after cancer cells undergo a very specialized process which is known as epithelial to mesenchymal transition. So they change from being epithelial cells to mesenchymal cells. They dissociate from neighboring cells, release various metalloproteases that degrade the underlying basement layer present under them, and then enter into blood circulation of the lymph system to spread to other parts of the body. So this is essentially what metastasis is. Is that fine? So here we have a diagram showing that we have an early stage tumor which changes to a late stage tumor, which releases cancer cells, degrading of basement membrane. They come into the bloodstream, they circulate throughout the bloodstream, and finally they can exit into another second location and infect a new site such as lung, liver, brain, or bone. So this is the process known as metastasis. Okay. And next we look at pluripotency. So pluripotency is not something that is related to cancer, but as we have discussed earlier, pluripotency is the describes the ability of a cell to develop into three primary germ cell layers of the early embryo. As we just saw, the three primary germ cell layers gives rise to the entire embryo, and pluripotent cells can give rise to these three primary germ cell layers, and thus the entire all the tissue types that are present in the embryo can be formed from pluripotent cells. Essentially, pluripotent cells involve the innermost cells of the blastula, which are known as embryonic stem cells. Pluripotent cells can form into all cells of the adult body, but not extraembryonic tissues such as the placenta. And embryonic stem cells and induced pluripotent stem cells are characterized by their pluripotency, by the ability to form every other tissue present in the body. So pluripotency is an attribute that is not connected to cancer cells, but is something entirely different. Is that fine?
Yes, ma'am.
Okay. So we have a diagram which shows ES cells, embryonic stem cells, which are pluripotent, followed by the three germ layers, which are multipotent, followed by omnipotent cells, unipotent cells, and finally fully differentiated cells. As we go down, we see a decrease in potency, that is the ability to form different types of cells. So pluripotent can form the most different type of cells, and finally we have unipotent stem cells which can form only one type of cells, and then fully differentiated cells which do not form any other type of cells. So the highly specialized process by which tumors escape the origin site and over time develop secondary tumor at a various other organ of the body is known as metastasis, and the attribute does not constitute a malignant tumor, that is, it is not associated with tumors or cancer. Out of all of the given four is pluripotency. Everything else, angiogenesis, invasiveness, genomic instability, are attributes of a cancer or a malignant tumor. So the correct option is metastasis and pluripotency. Okay, so that was that particular question. We move on to the last question that we have today, which is on Hardy-Weinberg law, which is the law which guides the using this law we can predict the frequency of various alleles in our large population. So we are given the following question: individuals with the wild type gene G is in humans make a glycoprotein. The mutant version of the glycoprotein works as a receptor for the seasonal flu virus, allowing it to enter the host cell. As a result, the person with the mutant gene becomes ill. A dominant allele produces the mutant glycoprotein capital G. So, if 84% of a population is vulnerable to the seasonal flu, that is individual with both the genotype capital G and capital G small g, what is the frequency of that mutant allele in the population, that is capital G. So this assumes the population is in stable Weinberg equilibrium. The options are given 0.16, 0.48, 0.6 and 0.8. So this is essentially a probability problem, not exactly probability but a mathematics problem using the Hardy-Weinberg law. So I would encourage you all to try and solve this on pen and paper. And go ahead and try. And if you are unable to do it, then we can do it using a whiteboard. But I will request you all to try and answer this first. Okay, so we have our first answer. So Shreya says that the option is C, that is 0.6. Okay, so but if we do not have any other answers, then I will go ahead and solve the problem, and then we can understand, like answer the question again. So I will need to present on another device. Yes. I hope the whiteboard is visible.
Yes, sir.
Okay. So we are given a problem of Hardy-Weinberg law. So first, let us. So what does the Hardy-Weinberg law essentially state? The Hardy-Weinberg law states that if the frequency of dominant allele in this case capital G is given by P, and frequency of recessive allele, in this case given by small g, is given by Q, then in a population which is under the Hardy-Weinberg equilibrium, we can draw the following statements from the law, which gives us that P plus Q equal to 1. This is number 1 point. And so if this is the first point. Secondly, if we go for the next generation, that is if we draw a cross between our homozygous recessive and homozygous dominant, so we have something that looks like no, not homozygous recessive homozygous dominant, but rather between two heterozygous population, we have, as we always know, that the structure will look, the Punnett square will look like this. So out of all of this, we know that capital G, if it denotes P, then or co-occurrence of two capital G's will be essentially P square. The heterozygous will be PQ together, another PQ together, and similarly, the homozygous recessive will be Q square. The law also states that in the F2 population, the following will also be equal to 1. That is, the distribution of the alleles will still remain as total to be 1 in the F2 population as well. These are the two parts of the law. P plus Q equal to 1 and P square plus 2PQ plus Q square equal to 1. As simple as that. Very easy. So essentially this law can be easily used to solve population genetic problems. So let us go ahead and understand what it is given. We are given that vulnerable to seasonal flu, the amount of population that is vulnerable to seasonal flu is essentially 84% or 0.84 of the entire population, the fraction of the frequency. So now, vulnerable to entire flu, to the seasonal flu, essentially is are the, as given in the question, the genotypes capital G and capital G small g, which is 0.84. Now we know from the previous Punnett square, right here, capital G can be denoted by P square. Capital G and small g are denoted by 2PQ because they can occur twice. So essentially, we have P square plus 2PQ equal to 0.84. This is by Hardy Weinberg law. Up to here, are we fine with this?
Yes, sir.
Okay. From here, we can solve it in three different ways. Like we essentially have a quadratic equation that we have to solve. So this is very easy to solve. So I will show you the three ways and you can choose whichever is the absolutely best you feel. The first one is you first substitute Q from this equation number 1. So P plus Q equal to 1. That means Q is 1 minus P is equal to 0.84. Bring it to a one quadratic equation with one variable. So we have 2p square plus p square plus 2p minus 2p square equal to 0.84, or we have p square minus 2p plus 0.84 equal to 0, which is essentially a lot of a big quadratic equation. So essentially from here on also you have here on you have two ways to solve this. First is you do it y. So this is just a simple quadratic equation, nothing else. So you just solve it either y by opening up the fraction. So 84 divided by 100 equal to 0, 25, or p square minus 50, 25 p square minus 50 p plus 21 equal to 0, and you then go ahead to solve this using the whatever technique you want. Otherwise, so you can solve this easily by the Aryabhatta method, that is you use p equal to minus. So the Aryabhatta, you can use this method to solve this, or you can be a bit smarter about this and you go from 2 you had. So this is the first method that you can solve it by just solving the quadratic, like brute force it, solve it by separating the middle term using the Aryabhatta method, whatever. The second you can use is you can just go ahead and write down the equation again. So this was what we started with. So this is something people who are savvy with maths can easily look at and understand, that this part of the equation looks pretty much like a perfect square. So essentially what we will do is that we complete the perfect square by addition of 1. So we have to add 1 to both ends. Okay, the plus was, wait, the signs are wrong. What were the signs? No, this was the signs. Okay, the signs are correct. Plus equal to plus, plus. And then we have P plus Q. Oh, no, wait. You add Q square to both ends. And then now it becomes a perfect square. P plus Q square equal to 0.84 plus Q square. So, but again by Harvey-Weinberg law, this P plus Q square is 1, as we saw right here. So we essentially have Q square plus 0.84 equal to 1, or Q square equal to 1 minus 0.84 equal to 0.16, or which is essentially Q equal to plus minus root over of 0.16, which will give you plus zero point 0.4 and minus zero point 0.4. But because the frequency cannot be less than 0, so Q is only 0.14, 8.4. And from this particular thing you can again use the equation P plus Q equal to 1, which will give you P as 1 minus Q equal to 1 minus zero point 0.4. Therefore P equal to 0.6. That is frequency of mutant dominant allele, capital G, is 0.6, which is option number C. Is that fine?
Yes, sir.
Okay, just a bit of mathematical juggling, and we will have it. So, going back to our presentation, yeah, I hope the screen is visible. Screen has changed. We go back to our PowerPoint. The PowerPoint is visible, correct? The PowerPoint is visible, right?
Yes, sir.
Okay. So here I have roughly solved the problem and given on the slide for you to refer back to. P plus Q equal to 1, P square plus 2PQ plus Q square equal to 1 is the Hardy-Weinberg law, and using that particular homozygous dominant plus heterozygous is 84%, P square plus 2PQ 0.84. Let's solve, just complete the perfect square. One side becomes 1. Then you just subtract and square root, and you then use again Hardy-Weinberg law to get back your frequency of dominant allele in the population. So P becomes 0.6. Our correct option is C. Okay. So any questions from this? No. Okay. So that was today's session. In this session we learned a lot about cell cycle dysregulation, developmental biology as well. Touched upon Hardy-Weinberg law. So this is the YouTube channel link on which all this recorded session will be uploaded. So if anyone else has any questions regarding the week's lecture or the course, I guess not. So I thank everyone for attending this PMRF NPTEL session, and see you all next week. Thank you.